Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My goal is to find interesting and exceptional researchers and clinicians and scientists and CEOs and interview them and ask them questions that they maybe normally wouldn't get and bring that information to you, the listener. So today I have Kellye Eversole. She's a pioneer in agricultural genomics, biotechnology, and information technology. She has two big initiatives that it appears she works with since 2005, the International Public/Private Consortium, the Wheat Genome. It's wheatgenome.org. We're going to be talking about that a bit, and then she's part of the Alliance for Phytobiomes Research, and that's phytobiomesalliance.org. So we're going to be talking about both initiatives. If I try to describe them, I'll mess them up, so it's better to ask her. So, Kellye, thanks for coming.
Thank you very much, Richard. It's a pleasure to have the opportunity to speak with you today.
Yeah, these, to someone that doesn't know what you do, these sound like two unusually specific things to work on. But, in your own words, why do you work on looking at wheat genomes, and what is this Phytobiome Alliance?
Sure. So the wheat genome effort was really a way to try to build the foundation for a new paradigm in wheat breeding. It takes on average 12 to 15 years to develop one variety of wheat. And part of that is because it takes a long time to grow out the wheat and find the right varieties to mix to then get the genes in. So one of the genomics was really high genome sequence. We could then bypass some of those years of breeding by using computational tools and really looking at the genes in specific varieties, and to do something that we call genome selection, genomic breeding. And one of the things to be able to do that is you have to have a high quality sequence that actually allows you to find the different genes. And as we've done and seen with the human genome, where we're really targeting some medications and pharmaceutical products, or even healthcare advice towards the genetic makeup of individuals, it's the same thing in wheat, where we really want to be able to say, okay, we have a drought problem, we want a drought resistant wheat, or we have a disease problem, we need a disease resistant wheat. So how do we make sure that disease resistant genes are in the variety that we are selecting? And so the genome sequence actually tells us which genes are in which varieties, and then we can increase the time that it takes or decrease the time that it takes to actually develop a new variety. And that's critical when you think about it, because if it takes 12 to 15 years to develop a variety or your target, by the time the disease hits and then you realize, well, we've got 12 or 15 years before we're going to have something to address it, then you just don't have the time to prevent it from wiping out 20 to 30% of the crop. And so that's really what the background of this was all about. And
quick sure. Question here. I don't know much about wheat. I've heard, legend has it, it's just one kind, maybe with a few variations, or just a few kinds. Like, what is the state of most of the wheat that people eat? Is it diverse or no?
There is quite a bit of diversity. There are a lot of different varieties that are out there, and varieties that are established for different parts of, in the United States, different parts of the country, or different parts of the world. Wheat is the most widely grown crop, and because it's the most widely grown, you do have a lot of different varieties. But in terms of, a lot of people have said that we have less genetic diversity than we used to. There's been those claims, but it's really not true. We have as much genetic diversity as we've ever had. That doesn't mean we don't need more, but we've always had quite a bit of genetic diversity. There's not just one variety of wheat that's grown. There's not even, there's wheat that's targeted for harvesting in the spring. There's wheat that's targeted for harvesting in the fall. So it really just varies.
And the Phytobiome Alliance, can you go into that briefly, and then we'll kind of ask questions back about both?
Sure. So in some regards, the Phytobiomes Alliance really was an outgrowth from an intellectual standpoint for me, because even though you might have the genome sequence of a plant, that plant, regardless of how much you know about that plant, is going to be influenced, and its growth is going to be influenced by what's in the environment. And we've heard a lot of talk over the years about, we want farmers and ranchers to do things in a sustainable way, and we make these assumptions what that means, and we have these broad sweeps that everybody needs to do a particular practice on their farm, and that therefore makes them sustainable. Well, the reality of it is it really depends on the specific site, the specific farm. I grew up in Southwest Oklahoma, and what works in Southwest Oklahoma may not work in the state of New York, for example, and may not work even in a field 100 miles away. So, there's really this need to understand what's going on in a particular field and then target things to that field and really give the growers all the tools that they need to be able to have sustainable production on their farm. And the whole concept of the phytobiome, it's a holistic systems approach. And it really, if you think about what the term means, phyto is simply a term for plant, and biome means a specific space. So like when we often talk about the human gut microbiome, we're talking about the microbes in a particular in the gut. So it's just literally related to the microbes in that human gut. In the phytobiome, we're talking about the plant in a particular environment or in a particular site. And so the concept is, can we begin to understand all the geophysical and biological components that impact and that affect the production of a plant in a particular field, of a plant on a rangeland, or even our forest, looking at our forest, what's going on in that forest that would allow us to have both the best genetics, what's the best crop genetics, what's the best crop, or what's the best tree species for a particular area, and what's the best management practices, when and what kind of nutrients do you put in the field or on the plant. So it's really trying to move towards what we call next generation precision agriculture. And if you think again about the movement in precision medicine, where we're trying to be able to target things specifically to individuals and have combine the genetic information with their own lifestyles and their activities, and design their medication packages based on that. In a similar way, we're trying to do that in agriculture, and to really figure out what's the best thing for a particular space. So that's the whole concept of the phytobiome. And if you think about it, it's really recognizing that we can't deal with things in a very reductionist manner. And it links with one of the things that just intellectually led me to it, was even thinking about the wheat genome, even though because it's so complex, and by the way, it's 5 times the size of the human genome, so it's really big.
Huh. Yeah.
And it's also a hexaploid, meaning that we have 6 copies of every chromosome. So it makes it really hard to deal with. Even if we understand that, and even if we perfect what we think is the so-called best wheat variety, it's only the best wheat variety for a place. And so how do you then go that next step? And those foundations that we've been able to build, both in terms of genomics and really computational tools and technologies that have come out in the past 10 or 15 years, is now allowing us to move into this bigger area that says, okay, now we can really look at everything and start trying to piece different things together.
So we're being reductionist in the way that we're engineering new wheat and let's say other plants, and then just expecting whatever engineering we do to pan out. But I guess what we're ignoring is, again, the microbial attachment to that particular plant and how that will change, and then epigenetic changes from the plant, being exposed to given environments and chemicals, and I guess a lot of things, right?
Yeah, exactly. I mean, and if you think about, and it's not just true in agriculture, but in science in general, even the way we train scientists generally has been to put them in narrow fields, narrow disciplines. And that's great if they're going to do nothing but teach that narrow discipline, and stay in academia and focus on that narrow discipline. The problem is if you want to get to application and you want to really look at the real world is not reductionist. It's extremely complex. And, it's the only way we can really understand it is to embrace that complexity and move beyond one discipline and think much, much more in a way of what they call transdisciplinary research and transdisciplinary activities, where what is our problem? Our problem is increasing sustainable production on a particular farm. Well, it takes agronomists, soil scientists, fertilizer specialists, plant physiologist, plant geneticist, and the farmer to be able to really think about what we need to do on that. So we have to bring all of that together, and that's where, we really need to move into embracing complexity.
What does that mean? We're engineering new types of wheat and other plants, but so right now, again, not reductionist. Like how can it be done in a smarter way? What's an example?
So one example would be is that, we really try to bring together, we look at a particular field and we look at a particular farm and we understand what's going on without a plant in that environment. So we understand the microbes, we understand what other insects or animals might be in that field at various times, what birds might be passing through, what is going on in terms of the environment or wind erosion, et cetera. So you really have the understanding of the weather. And then you try to bring all the different components into one research project where you're beginning to look at the network of interactions. So you tweak one aspect. For example, maybe you want to change the microbial community that's in a particular field, because you can now put microbes in that would make the plant or make any plant or a particular plant have more uptake of nitrogen, for example. And so you can't do that in isolation. You've got to do that. Okay, what if I do add these microbes? Then I need to understand what that does to the plant itself, to the physiology of that plant. So you've got to have all of those disciplines at the table. And that's really what we're trying to do within the Phytobiomes Alliance, is to get all of these disciplines to talk to each other and to realize that we need to work together to solve these problems. And we don't need, for example, just to understand what the physical components of soils might be, which tends to be where the soil scientists go. We need to understand the microbial communities there, what treatments or management practices a grower might be using in any given year, et cetera.
So what's an example of doing it right or doing it wrong, and what is the result? No yield, low yield. How do you know when you're doing things the right way? What has to be done?
Well, we're just now getting started in this, and so one of the greatest challenges is even to begin to see how many different components we can actually bring together at one particular time. But one impact might be we can substantially reduce the amount of runoff for pesticide runoff, fertilizer runoff, et cetera, on a particular farm. And so to improve the environmental sustainability of a farm just by tweaking the plant genetics and combining it with maybe what a microbial product that could be added to the soil. We are just getting started, so we don't have concrete examples yet, but there are a lot of promising areas that are starting to surface, even from a standpoint of understanding whether on a particular farm for a number of years and seeing what those patterns would be.
So is this experimentation taking place? And, is there any interesting observations about, when a new genetically modified plant is on the scene, what has been observed that was intended and unintended? Any examples?
I'm not really talking, when I talk about genomics, I think there is this idea that in agriculture that means genetically modified crops, and that's not necessarily what I'm talking about. You can use genomics and use the tools of genomics to create crops in a conventional manner. It just shortens the time that it takes to do that. In terms of impacts, we've been seeing, a lot of impacts in terms of genomic breeding, primarily in crops that have had a genome sequence for a number of years, such as rice, maize. There has been of the time it takes to go from discovery to actual commercial release of hybrids or varieties. But, I guess I don't really completely understand what you mean in terms of it, unintended effects or unintended.
Yeah, they'll again, certain plants or crops have been engineered, and then I don't know if you've observed when they've first gone into production in someone's field, and what things happen that were intended, what things happen that were unintended, or inexplicable.
Yeah, so it's not necessarily it's not that it's inexplicable. It's more that because of the specific environment that something is in, a variety, for example, that might get as much as 100 bushels per acre might not get 100 bushels per acre on a particular farm. And so and then you have to figure out, well, why is that the case? What is the, were the management practices different? Did the weather have an impact? Did you have a disease problem? Or did you have an insect outbreak that impacted your variety that year? So you really have to understand it from those perspectives. Or in some cases, it might be that simply a farmer's field might be has different microclimates. For example, and that might actually be because of the nature of that field. It might be a part of it might be surrounded by trees. So you might have a small microclimate on one end of a field. The other aspect that becomes, and that we're learning more and more about, is the fact that the soil is very different. Even if you have an open field, for example, my small farm in Oklahoma has 3 different soil types in that field. And so that in itself is going to impact that variety. And right now, the only thing that we're able to do is to really look at how many different varieties are available on the market, and then you pick one and you plant it across the whole field, but you're going to get different results depending on the field itself. And that's what, with the Phytobiomes effort, we're really trying to get an understanding of how do we really target for a particular field and the different parts of that field. And where we've seen the most advances in understanding this site-specific agriculture is in the area of fertilizer, and really applying specific amounts of fertilizer to a particular acre of land, for example. And that's built up over time because we have data. We have, maybe in some cases they have 10 or 15 years of data with the grids of that particular acre and how it is impacted by different levels of fertilizer. And so you really try to, what's the right amount of fertilizer for that particular acre of land, and what's the right application rate, what's the right time that you want to put it on, and that will ensure that you have a higher sustainability, less runoff, et cetera.
And that's interesting. I guess, you might think a field of plants is just a monolithic thing, but you're right, there's different microclimates. Bugs may visit part of a field and not other parts. The angle of the sunlight, the soil beneath them, you treat the field with a given fertilizer because of the elevation or the gradation of the underlying dirt. It may run from one place to another preferentially, and and pool. I mean, I guess there's tons of factors that would affect it.
That's exactly right. And in the past, we've not really been able to capture that. One, we didn't have the tools to capture it, and not the tools that would be cost effective. But secondly, we just didn't have the computational ability that even if we could get all the data, we couldn't manipulate the data and figure out, well, what's really going on here. And that's where we are today, is we have those kinds of opportunities now. And I think, the advancements that have been made in machine learning and artificial intelligence have really opened up a complete new field for looking at agriculture in a very different way, and really thinking about not only a farmer's field, but your garden. What does your garden look like? What should you do with your garden? And I know there's, there, particularly these days in the time of that, people are producing a lot or planting a lot of their vegetable seeds. Right now, I read somewhere there's been an increase in the production or in the sales of vegetable seeds in the past month, thanks primarily, I think, to COVID-19. But how do you, as a homeowner, how do you actually plant your garden? And how do you just go to your local garden store and ask him or her or the person at the garden store what you should put on your garden? But that person has no knowledge of what kind of a garden you have. What is that in your soil? What kind of soil is it? What kind of slope do you have? I mean, there are just a lot of different factors that impact the production of a particular plant. And that's what we want to get a handle on, and we want to empower the ability for anyone to be able to know that. And one of the areas and this is where kind of the genomics piece comes in, one of the areas that is the most exciting is that we're now seeing the ability to understand what microbes are in a field. What is there now? And, when we first started in this area about 15 years ago, no one thought we'd be able to do genome sequencing of a particular soil, because the number of microbes are astronomical compared to the human. There's like 70 to 80,000 microbes in any given soil sample. So how do you even deal with that? But now you can, because the cost has gone down, the computational capacity is there, and we can finally begin to develop the information that we need to change how we think about agriculture and change how we produce.
Has it been observed that there's a plant-to-plant communication in a given field?
Yes.
If you do something to plants on one side of the field, that the effect will be seen on the other side?
Yeah, there is. We do know there's communications, and that's one other aspect of the area of phytobiome, is to understand what other plants might be in the area. For example, what weeds might be in the area. What in some cases, farmers plant what they call buffer strips, where it's a plant to try to hold the soil in or to filter environmental contaminants. What impact is that having on a particular plant? And, that we don't really know. Some of the plant-to-plant communication that they're just now beginning to understand is simply the same variety, but it's in a different field, and it's because of the pollination method.
So how do you go about tackling optimizing a field, or at least, I guess, first is putting sensors or looking at a field, sequencing the soil, like you said. So if you're going to have a project, what would that look like? Would you just have you start with a planter box, or like a real small area, maybe a 10-foot by 10-foot plot, and see what you can do to optimize that plot? Like how would you experimentally carry this out?
Yeah, I think it really depends on the field itself. It's one of the questions we get and are trying to deal with all the time is, how much of a field do you need to sample to get a good idea of what's in the field? And that's true whether it's just straight physical characteristics of soil, or whether you're actually also doing the microbial sequencing and trying to understand the metagenomic communities that are in that field. And so I think right now what we're trying to do is to try it at different levels. We're looking at, small plots to very large fields. And by large fields, we're talking 5 or 6 acre fields. And there are different trials going on in different parts of the world where they're trying to see if there is a difference. Right now, the cost is not low enough for us to do it on every acre or really to get to drill all the way down even to a square foot level. But, that the goal is, do we really have to get there? Can we develop some models that would allow us to then know that if we can do it on 20% of the field in a random selection, and that will give us a good enough picture that we can actually use. And the same is true for weather, bringing in the climate impacts. How many years of weather do we need to look at to begin to understand what the patterns might be for that particular field, and what impact the weather had on different years of production.
So any results from experimentation that's been going on already where you're seeing interesting differences in a field?
Not really yet, because we're not sampling a broad enough pool yet. We're still quite a bit of ways from actually having that. We do know that we are seeing tremendous differences already in fertilizer, starting to see differences in what we call seeding rates, where you can actually put less seeds down in a particular acre than you would in another acre because it grows better or it doesn't grow as well. So you're changing that. So that kind of activity is already going on. And there is, I think some of the companies are beginning to look at microbial products and that would go onto a field that would increase the nitrogen uptake or would increase the drought resilient, for example. And from my understanding on some of those, they're beginning to get an idea of exactly how much they need to sample a particular field before they put their products down.
So what do you particularly have any projects in the works right now that are going to be involved in figuring out what's going on with the fields, or are you more of an overseer of what's happening?
So we've got some projects that we're developing at this point. One that we're putting together right now that would be an international project, and it's really will take us all the way from breeding to adding different microbial products to particular vegetable and legume crops, and that will give us some kind of an understanding of what kind of biological products might be useful to help with the nutrients of the particular crop, for example, health benefits of a particular crop. And if we get that one funded, then that'll be our first major one that really tries to do it at an international level. There are others who are doing, not me personally, but within our consortium, who are doing different components of the phytobiome. A lot of it has been focused on trying to understand the microbial communities in agricultural fields. And so that is going on. Others are working more specifically on trying to combine nutrient use with the microbes and the plant. And so there are pieces that are starting to get put together, but not that I am particularly involved in. Really what we do is I'm usually leading one particular or a PI, a co-PI on one or two particular projects. But overall, we're trying to increase the collaboration and increase the coordination among a variety of projects internationally.
So what's the best way for people to find out more about the initiatives and keep an eye on the learnings that are going to come from this?
So we have a couple of websites. I think you mentioned them in the beginning for the Wheat Genome Project, which is continuing, and we're trying to drill down more and even expand the diversity in wheat. You can find information at www.wheatgenome.org. And for the Phytobiomes Alliance, that is website is www.phytobiomesalliance.org. And then for generally to see a number of the projects in which I'm involved and those on my team are involved, we also have a website at www.eversoleassociates.com.
Okay, well, very good. Well, Kellye, thank you for coming, and I wish you knew now, but we'll have to revisit when you get some more juicy details on what happens and how much yield can increase and how much more understanding can come from
Yeah, so we do have, there is some results, at least from what people are reporting. Some of the science publications have shown that you can get 5 or 10% increase in yield by using specific biological products that enhance the microbial communities in a particular field. But again, that's just looking at a couple of different components. It's not bringing in, the full scope. But, that takes time, as you well know from your work and focus on a lot of the biomedical.
Well, very good, Kellye. Thank you for coming on the podcast today. I appreciate it.
Well, thank you very much, Richard. It's been a pleasure.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.